Metastasis is a complex process where cancer cells break away from their original site and spread to other parts of the body. This ability to disseminate is what makes cancer particularly dangerous and difficult to treat. In this video, we'll explore the fascinating mechanisms that allow cells to escape their primary location, travel through the body, and establish new tumors elsewhere. Understanding these mechanisms is crucial for developing better therapies and improving patient outcome. Let's begin by looking at what metastasis really means and why it's so significant in cancer biology. Journey of metastasis starts with the formation of a primary tumor. This is where abnormal cells begin to grow uncontrollably, forming a mass in a specific tissue or organ. These cells often acquire mutations that allow them to ignore normal growth signals and evade the body's defense mechanisms. As the tumor grows, it creates a microenvironment that supports its survival and expansion. This environment is rich in nutrients and growth factors, setting the stage for the next steps in the metastatic process. Cancer cells to leave the primary tumor, they must undergo significant changes. These changes are often triggered by genetic mutations and signals from the tumor microenvironment. One key transformation is called the epithelial mesenchymal transition, or EMT. During EMT, cells lose their tight connections with neighboring cells and become more mobile. This increased mobility is essential for the next phase of metastasis, as it allows cells to break free from the tumor mass. Epithelial mesenchymal transition, or EMT, is a process where epithelial cells acquire characteristics of mesenchymal cells. This means they become less adhesive and more capable of movement. EMT is driven by various signaling pathways and transcription factors. It's a critical step in metastasis because it enables cancer cells to detach from the primary tumor and invade surrounding tissues. Without EMT, most cancer cells would remain trapped in their original location. Major hallmark of metastatic cells is their reduced adhesion to neighboring cells. Normally, cells are held together by proteins like E-cadherin, which act as molecular glue. During metastasis, the expression of these adhesion molecules decreases, making it easier for cells to break away. This loss of adhesion is a direct result of EMT and is essential for the initial steps of cell dissemination. Once cancer cells lose their adhesion, they begin to invade the surrounding tissue. This invasion is facilitated by enzymes called matrix metalloproteinases, or MMPs, which break down the extracellular matrix. By degrading the structural barrier, cancer cells can move more freely and penetrate deeper into nearby tissues. This invasive behavior is a defining feature of metastatic cancer. After invading the surrounding tissue, cancer cells must enter the bloodstream or lymphatic system to travel to distant sites. This process is known as intravasation. Cancer cells squeeze through the walls of blood vessels, often with the help of signals from nearby stromal cells and inflammatory molecules. Once inside the circulation, they face new challenges, but are now capable of reaching almost any part of the body. Bloodstream is a hostile environment for cancer cells. They are exposed to immune cells, physical stress, and the risk of being destroyed. To survive, metastatic cells often travel in clusters or attach to platelets, which help shield them from immune attack. Some cells also express proteins that protect them from apoptosis or programmed cell death. These survival strategies are crucial for successful dissemination. While in circulation, cancer cells must avoid detection and destruction by the immune system. They achieve this by expressing molecules that suppress immune responses or by hiding within clusters of normal cells. Some metastatic cells even release factors that create an immunosuppressive environment, making it harder for immune cells to attack them. This ability to evade the immune system is a key reason why metastasis is so challenging to treat. After surviving in the bloodstream, metastatic cells must exit the blood vessels to colonize new tissues. This process is called extravasation. Cancer cells adhere to the vessel wall, squeeze through the endothelial layer, and enter the surrounding tissue. This step is often guided by signals from the target organ, which attract the cancer cells and help them settle in a new environment. Once cancer cells have exited the bloodstream, they must adapt to their new environment in a distant organ. This process, known as colonization, is often the most difficult step for metastatic cells. They must survive in a foreign tissue, evade local immune responses, and establish a supportive microenvironment. Only a small fraction of disseminated cells succeed in forming secondary tumors. Before cancer cells arrive, some organs create a premetastatic niche, a specialized environment that makes it easier for metastatic cells to settle and grow. Tumor-derived factors and exosomes travel through the bloodstream to distant sites, where they recruit supportive cells and modify the local tissue. This niche formation is a key factor in determining where metastases are likely to develop. For metastatic tumors to grow, they need a blood supply. Angiogenesis is the process by which new blood vessels form, providing nutrients and oxygen to the tumor. Cancer cells release signals that stimulate nearby blood vessels to sprout new branches. This not only supports tumor growth but also facilitates further dissemination of cancer cells. Tumor microenvironment plays a crucial role in metastasis. It consists of various cell types, signaling molecules, and structural components that interact with cancer cells. These interactions can promote cell survival, invasion, and dissemination. Understanding the microenvironment is essential for developing therapies that target not just the cancer cells, but also the supportive network around them. Cancer cells, known as cancer stem cells, have a unique ability to self-renew and initiate new tumors. These cells are thought to play a major role in metastasis because they can survive harsh conditions and adapt to new environments. Targeting cancer stem cells is a promising strategy for preventing metastasis and improving treatment outcomes. Metastatic cells often acquire additional genetic and epigenetic changes that enhance their ability to spread and survive. These changes can affect gene expression, cell signaling, and interactions with the microenvironment. By understanding these alterations, researchers hope to identify new targets for therapy and develop more effective treatments for metastatic cancer. Exosomes are tiny vesicles released by cancer cells that carry proteins, RNA, and other molecules. They play a key role in communication between the tumor and distant organs, helping to prepare the pre-metastatic niche and promote metastasis. Exosomes can also suppress immune responses and enhance the survival of metastatic cells. Cancer cells use various mechanisms to move through tissues. They can crawl along extracellular fibers, squeeze through tight spaces, and even use amoeboid movement to navigate complex environments. These motility strategies are powered by changes in the cell's cytoskeleton and are essential for successful dissemination. Proteolytic enzymes such as matrix metalloproteinases are crucial for breaking down the extracellular matrix. By degrading these barriers, cancer cells can invade surrounding tissues and access blood vessels. Inhibiting these enzymes is a potential strategy for limiting metastasis. Cancer cells are guided by chemical signals or chemotactic cues that direct their movement toward blood vessels or distant organs. These cues can come from the tumor microenvironment or from distant tissues that are preparing to receive metastatic cells. Understanding these guidance mechanisms could lead to new ways to block metastasis. Stromal cells in the tumor microenvironment, such as fibroblasts and immune cells, play a supportive role in metastasis. They secrete factors that promote cancer cell survival, invasion, and migration. Targeting the interactions between cancer cells and stromal cells is an area of active research in cancer therapy. Chronic inflammation in the tumor microenvironment can promote metastasis by providing growth signals and facilitating tissue remodeling. Inflammatory cells release cytokines and enzymes that help cancer cells invade and disseminate. Managing inflammation is therefore an important aspect of cancer treatment. In addition to the bloodstream, cancer cells can spread through the lymphatic system. Lymphatic vessels provide a pathway for cells to reach lymph nodes and eventually enter the circulation. Lymphatic spread is a common route for many types of cancer and is often associated with a worse prognosis. Normally, cells that detach from their original tissue undergo a form of programmed cell death called anoikis. Metastatic cancer cells, however, develop resistance to anoikis, allowing them to survive while traveling through the body. This resistance is a key adaptation that enables successful dissemination. Some disseminated cancer cells can remain dormant for years before forming new tumors. These dormant cells evade detection and are resistant to many therapies. Understanding the mechanisms of dormancy could help prevent cancer recurrence and improve long-term outcomes for patients. Metastatic cascade refers to the series of steps that cancer cells undergo to spread from the primary tumor to distant sites. These steps include local invasion, intravasation, survival in circulation, extravasation, and colonization. Each step presents unique challenges and opportunities for therapeutic intervention. Certain cancers tend to metastasize to specific organs, a phenomenon known as organotropism. For example, breast cancer often spreads to the bones, while colon cancer frequently metastasizes to the liver. This preference is influenced by factors in both the cancer cells and the target organs. Adhesion molecules on the surface of cancer cells help them interact with blood vessel walls and target tissues. These molecules, such as integrins and selectins, play a key role in extravasation and colonization. Targeting adhesion molecules could help prevent metastatic spread. Metastatic cancer cells often undergo metabolic changes that allow them to survive in different environments. They may switch to alternative energy sources or increase their ability to handle oxidative stress. These adaptations help them thrive in foreign tissues and resist therapy. Epigenetic changes, such as DNA methylation and histone modification, can alter gene expression without changing the underlying DNA sequence. These changes play a significant role in enabling cancer cells to adapt and disseminate. Targeting epigenetic regulators is an emerging area in cancer therapy. Low oxygen levels or hypoxia in the tumor microenvironment can promote metastasis. Hypoxia triggers the activation of genes that help cancer cells survive and invade. It also stimulates angiogenesis, providing new pathways for dissemination. Effective communication between cancer cells and their environment is essential for metastasis. This communication occurs through direct contact, secreted factors, and extracellular vesicles. Disrupting these communication pathways could help block the spread of cancer. Platelets in the blood can protect circulating cancer cells from immune attack and help them adhere to blood vessel walls. This interaction increases the chances of successful extravasation and colonization. Targeting platelet cancer cell interactions is a potential strategy for limiting metastasis. Mechanical forces, such as fluid shear stress in the bloodstream, influence the survival and behavior of metastatic cells. Some cancer cells adapt to these forces by altering their shape or reinforcing their cytoskeleton. Understanding these adaptations could lead to new approaches for preventing metastasis. Growth factors are signaling molecules that promote cell proliferation, survival, and migration. In the context of metastasis, cancer cells often produce or respond to growth factors that enhance their ability to spread. Blocking these signals is a key focus of targeted cancer therapies. Treating metastatic cancer is challenging because of the diversity and adaptability of disseminated cells. These cells can evade therapies, survive in different environments, and develop resistance to drugs. Overcoming these challenges requires a deep understanding of the mechanisms of dissemination. Researchers are actively investigating new ways to block metastasis. This includes targeting the molecular pathways involved in EMT, invasion, and immune evasion. Advances in genomics and single-cell analysis are providing new insights into the behavior of metastatic cells. Emerging therapies for metastatic cancer include drugs that target specific signaling pathways, immunotherapies that boost the body's defenses, and agents that disrupt the tumor microenvironment. Personalized medicine approaches are also being developed to tailor treatments to individual patients. Detecting metastasis early is critical for improving patient outcomes. Advances in imaging, liquid biopsies, and molecular diagnostics are making it possible to identify disseminated cells before they form secondary tumors. Early intervention can help prevent the spread of cancer and improve survival rates. Living with metastatic cancer presents unique challenges for patients and their families. Understanding the mechanisms of dissemination can help patients make informed decisions about their care and treatment options. Support from healthcare providers and loved ones is essential throughout the journey. Future of metastasis research is promising, with new technologies and approaches on the horizon. Advances in single-cell sequencing, imaging, and computational modeling are providing unprecedented insights into how cancer cells disseminate. Collaboration between scientists, clinicians, and patients will be key to translating these discoveries into better treatments. To summarize, the dissemination of cancer cells during metastasis involves a series of coordinated steps: loss of adhesion, invasion, intravasation, survival in circulation, extravasation, and colonization. Each step is driven by specific molecular and cellular mechanisms, offering potential targets for therapy. Understanding the mechanisms of cell dissemination has important clinical implications. It can guide the development of new diagnostic tools, inform treatment strategies, and help predict patient outcomes. Continued research in this area is essential for advancing cancer care. While metastasis remains a major challenge in cancer treatment, ongoing research and innovation offer hope for the future. By unraveling the mechanisms of cell dissemination, scientists are paving the way for more effective therapies and better outcomes for patients worldwide. In conclusion, the mechanisms of cell dissemination during metastasis are complex and multifaceted. From genetic changes to interactions with the microenvironment, each step presents unique challenges and opportunities. By deepening our understanding of these processes, we can move closer to preventing and treating metastatic cancer more effectively.